Have you ever wondered how the cells adapt to environmental stressors? Or how do they manage to regulate which genes are expressed and at what levels? Hi, welcome to my channel where I explain basic science processes in the simplest way. DNA is like an ultimate guidebook or manual that contains all the instructions in making proteins. But here's the twist: DNA never leaves the nucleus, while translation or the process of making proteins happens in the cytoplasm. So, how do you get those instructions from the nucleus to the cytoplasm? That's where transcription comes in. Transcription is part of the central dogma of molecular biology. It states that genetic information flows in one direction only, from DNA to RNA through the process of transcription, and RNA to protein through the process of translation. The process of transcription differs between prokaryotic and eukaryotic cells. In prokaryotes, the transcription happens in the cytoplasm where it is often coupled with translation, meaning transcription and translation happen simultaneously. In prokaryotes, functionally related genes are clustered together in a single unit called operon, which can be transcribed together into a single mRNA molecule. Operon is composed of promoter, an operator, and structural genes. The promoter region has two major landmarks, the -35 box and Pribno box, which serves as signals where the RNA polymerase will bind, just before the +1 site or transcription start site. The RNA polymerase, which is a multi-subunit enzyme, will bind to the promoter region. The RNAP is guided by the sigma factors to locate the specific promoter sequences like -35 box and Pribno box. The sigma factors decide which sets of genes are turned on. When a bacterium is under stress, it can switch sigma factors, thereby shifting its gene expression to stress response genes. The RNAP unwinds about 14 base pairs around the Pribno box, creating a transcription bubble. The RNAP will then perform DNA scrunching, wherein it will pull downstream DNA to itself. This will serve as a spring, allowing the RNAP to move forward and proceed to elongation. The RNA positions the incoming nucleoside triphosphates opposite the DNA template base. The selection is based on the Watson's-Crick base pairing rule, where adenine pairs with the uracil and cytosine pairs with the guanine. The RNAP moves along the DNA template, elongating the nascent mRNA. Termination in prokaryotes occurs in two ways: raw-dependent and raw-independent termination. In raw-independent termination, it relies only on the RNA sequence being transcribed. The presence of GC inverted repeats form hairpin loop structure acts as a physical obstruction for transcription. Moreover, the presence of polyuracil forms weak bonds which can be easily disrupted. The combined tension from the hairpin loop structure and the weakened uracil adenine pairing causes the mRNA transcript to detach and fall off the RNAP, thereby terminating the transcription. On the other hand, the raw-dependent termination relies on the raw protein, a hexameric ATP-dependent RNA helicase. Raw binds to the raw sites of the nascent RNA transcript and uses ATP hydrolysis to translocate, chasing the RNAP. The raw protein catches up and unwinds the RNA-DNA hybrid using its helicase activity, which causes the release of RNA and the termination of transcription. In eukaryotic cells, the transcription happens in the nucleus, wherein it will undergo post-transcriptional modification before it gets transported to the cytoplasm for translation. In eukaryotic cells, the gene contains introns and exons. The exons are the coding regions of the genes, while the introns are the non-coding regions which will be spliced later on to transform the pre-mRNA into mature mRNA. The eukaryotic gene contains the promoter region, which contains the total box wherein the general transcription factors will bind. The Tata binding proteins detects and binds to the Tata box. This will recruit other GTFs to the region, which ultimately lead to the recruitment of RNAP2. The name RNAP2 is mainly due to the presence of other RNA polymerases, but only RNAP2 transcribe all protein coding genes into mRNA. The transcription starts on the transcription start site. However, the transcription will not begin without the distal regulatory elements. Upstream, farther away from the promoter, where the regulatory elements like enhancers are located. The regulatory elements influence the transcription of the gene. The specific transcription factors will bind to this region, which leads to the recruitment of various proteins that leads to the looping of the DNA. This will bring the distal regulatory elements closer to the promoter, thereby efficiently influencing the transcription. One of the GTFs called TF2H has helicase activity, which unwinds the DNA helix, allowing transcription to begin. It also contains kinase activity, phosphorylating the RNA-P2, thereby activating it. RNA-P2 now starts moving along the DNA template, synthesizing RNA. The transcript is cleaved at an internal site before RNA-P2 finishes transcribing. This cleavage site is considered the end of the gene. The remainder of the transcript is digested by a 5' exonuclease, while it is still being transcribed by the RNA polymerase II. When the 5' exonuclease catches up to the RNA polymerase II by digesting away all the overhang RNA, it helps disengage the polymerase from its DNA template strand, finally terminating that round of transcription. Now, the newly transcribed eukaryotic mRNA undergoes post-transcriptional modifications, the 5' capping, splicing, and polyadenylation. The 5' capping is the modification that occurs at the 5' end of the RNA. Specifically, a modified 7-methylguanosine cap is added at the 5' end of the RNA, which protects it from the degradation of exonucleases. Moreover, the cap also forms the binding signal for cap binding proteins for translation. The cap binding proteins are important for nuclear export and translation initiation. Remember that the encoded RNA still contains the introns or the non-coding regions, which must be removed. The process of splicing is facilitated by spliceosome, a complex of small nuclear ribonucleoproteins and proteins. Spliceosome recognizes and binds to the consensus sequences such as 5' splice site or the GU motif and the 3' splice site or the AG motif, the polypyrimidine tract, and the branch site A. First, the 2' hydroxyl group of the branch point A performs a nucleophilic attack on the phosphate at the 5' splice site. The intron is covalently linked to the branch point A, forming a lariat structure loop. Then, the 3' end of the exon now has a free 3' hydroxyl group, which will attack the phosphate at the 3' splice site. The exons are now ligated together by a normal 3' and 5' phosphodiester bond, while the intron lariat is released and will be subjected for degradation. Polyadenylation is the addition of poly-A tail of an ascent pre-mRNA. This process starts with the recognition of polyadenylation signals by the cleavage and polyadenylation specificity factor or the CPSF. Downstream of this signal is the GU-reach or the U-reach region or the actual cleavage site where the cleavage stimulation factor or the CSTF binds. The CPSF and CSTF work together to ensure that the cut happens at the right spot. The CPSF73, an endonuclease enzyme, actually performs the cut in the pre-mRNA. The poly-A polymerase functions to add a long string of adenines of about 200 to 250 adenine nucleotides at the new 3' end to form the poly-A tail. After all of these post-transcriptional modifications are done, the mature mRNA is now ready to be transported into the cytoplasm for the translation process to begin. Again, transcription is the process by which the genetic information stored in DNA is transcribed into mRNA. It begins when RNA polymerase binds to a promoter region on the DNA. Once bound, the enzyme unwinds the DNA and uses one strand as a template to assemble a chain of RNA nucleotides, matching them to their complementary DNA bases. Eventually, Termination signals in the DNA sequence allow the termination of the transcription. In eukaryotic cells, the transcript undergoes further modifications such as splicing, capping, and polyadenylation before becoming mature messenger RNA. Now that we learned about the process of transcription, its difference between prokaryotic and eukaryotic cells, as well as the post-transcriptional modifications done to nascent mRNA to transform it into mature mRNA, I hope you'll appreciate even more the complex processes done by our cells in our day-to-day lives. So again, knowledge is power, so always keep that spark alive. So until the next video, see you!